So we're going to go ahead and go back to the PowerPoint and talk about the resting potential. So the cell membrane's function is to separate what's outside the cell, mostly sodium, what's inside the cell, potassium. So we also have negative sodium right here. You can see the sodium. There are leak channels where, since there's an abundance of potassium in the cell, allows potassium to leak out, sodium to leak in, but overall the inside of the cell is slightly negative with respect to the outside. This sodium-potassium pump, notice it pumps potassium in against a large gradient, meaning that this positive charge repels these potassiums being pumped in by the sodium-potassium pump. So there's a high concentration of potassium in the cell. There's the positive charge of the potassium, so this pump has to have energy to pump potassium in and to pump sodium out. So this sodium-potassium pump maintains excess sodium on the outside of the cell and excess potassium inside the cell, with the general effect of creating a negative charge on the inside of the cell based upon all these negative proteins which cannot move because they're too big. They cannot move in or out of the cell. So a voltmeter will record a voltage. A voltage is a difference from one area to the other with the relative amount of positive and negative charges. So the membrane potential can change with movement of ions. Ions move in channels that cross the entire membrane. They can open or close to allow sodium in. That causes depolarization. So if sodium comes into the cell, the potential or voltage will change to become more positive. If potassium channels leave the cell, then the potassium moves out of the cell and causes hyperpolarization, meaning that the voltage of the cell returns to a more negative voltage compared to the outside of the cell. So the membrane potential exists because of the ionic composition. The cell membrane allows certain ions like sodium and potassium to move through channels, and this potential can change as the cell first allows sodium, then allows potassium out. So when you have somebody pressing on your skin, for instance, you can feel that because receptors in the skin will respond to pressure, and they'll begin to send messages to your spinal cord and then up to your brain. So the action potential depends on reaching a certain voltage called a threshold. First of all, sodium channels allow entry of sodium. That causes the voltage inside the cell to become more positive. Then potassium channels open. Potassium leaves, causing the inside of the cell to return to a negative voltage. Then the refractory period is when the nerve has to recover and be ready for another action potential. So if we look at a diagram, you can see that there's sodium, excess sodium on the outside. There's excess potassium on the inside of the cell. When a voltage change becomes high enough to open up enough sodium channels from sodium entering based upon a neurotransmitter opening channels in the dendrites. So the first thing that happens is this sodium channel, which has an opening toward the exterior and another opening in the cell. Sodium's going to rush in. That sodium's going to rush in, and the sodium rushes in as the membrane potential goes from -70 to +35. The sodium's open up very much, and then the potassium channel's opening with the closer of this internal gate on the sodium channels. Once all of this potassium rushes out, we have hyperpolarization, and during that period, one nerve impulse moves down the axon away from the cell body, and we get ready for another nerve impulse with the sodium channel closed and the potassium channel closed, and the sodium potassium pump is routinely pumping sodium out and potassium in. So there's two types of movement of the action potential. So propagation or movement of the nerve impulse, there's continuous propagation and saltatory propagation. So continuous means in non-myelinated axons, once there's a stimulus at the dendrites by a neurotransmitter being released, there's sufficient sodium that comes in first. That sodium moves toward this negative charge right at this membrane interface, and you can see that the sodium is going to move as a local current, as it's called, and that carries the impulse down the nerve, meaning there's actually a change in sodium coming in and flowing forward away from the cell body. This movement of sodium in happens first, and then the potassium will go out, but that electrical change in voltage, gets to the synapse and can release neurotransmitters such as acetylcholine, dopamine, norepinephrine. So nerves carry spikes or nerve impulses by changes in movement of ions into the nerve membrane, through the nerve membrane, and those changes in voltage will lead to the release of neurotransmitters. For instance, at the muscle, we learned the neurotransmitters can cause channels to open, and that's a biochemical stimulus that allows sodium to go into the cell. Okay, so if you have a myelinated axon, you can see you have this fatty myelin tissue which stops any movement into the cell membrane, through the cell membrane into the cell, and the sodium can only move in at these gaps called nodes of Ranvier. So here you have the myelin. So the local current means the movement of positive charge in, and that current quickly moves to the part of the cell where it's negative and there's no insulation. So the saltatory conduction means that the impulse jumps from node of Ranvier to node of Ranvier. You can see that as the inside becomes positive with the movement of sodium in, you can have channels open at the node of Ranvier, and those local currents with sodium coming in moves down the axon away from the cell body. Behind that is the restoration of the nerve voltage back to around -70. But impulses routinely move away from the cell body toward the axon ending. Okay, more about that. So information travels away from axons down to axon endings. These electrical signals are known as action potentials. They carry information about vision, balance, movement. It's carried by myelinated axons. So Every time we move, our muscles, myelinated axons are involved. And when you are looking at this screen and you're able to read information travels within the nervous system in these electrical signals known as action potentials. They're also known as nerve spikes and nerve impulses, and they always travel away from the cell body down the axon, and they happen because of sodium channels are open and the sodium rushes in, causes the voltage to become more positive. Then as that happens, the sodium channels close and then the potassium channels open and potassium leaves the cell, returning the cell to its negative voltage. Okay, what are synapses? Let's stop there. We'll get to synapses. We'll stop recording, stop recording.